Welcome back, friends. In this video tutorial we will going to talk about complement fixation. So complement fixation, before going to talk about this, it is necessary to know about complement. So complement is a immune system protein, a protein which is involved in our immune response. Okay, and these proteins are all the time normally present in our serum in inactivated form. Okay, now when it is required, they become activated. Okay, and these proteins are globulin in nature. First thing is this protein is globulin in nature. And here remember, three type of complement pathways are available. One is classical complement pathway, second one is alternative pathway, and the last one is lectin pathway. So here the complement fixation step relies on the classical pathway of complement fixation, okay, which involve antigen antibody complex. Okay, so whole complement system is made up of 9 components, okay, from C1 to C9. It includes C1, C2, C3, C4, C5, C6, C7, C8 and C9. And these proteins are heat labile. They can be destroyed by heating at 56 degrees centigrade for 20 to 30 minutes, a process called inactivation. Okay, so the idea is, suppose this is a cell and on the surface of the cell there are antigen present. Okay, let's say this. Okay, I cannot zoom it. Okay, I can do this. So let's say these parts are antigens which are present on the surface of a cell. Let's consider this one as a bacterial cell. Okay, now there will be specific antibody for that particular antigens, right? Okay, now the complement proteins will bind over here, okay, and facilitates the lysis of that cell via membrane attack complex formation. Okay, that is the overall idea of classical complement pathway. Okay, if you want to know more about complement system, please let me know in the comment box. I will surely try to make a video on complement system. Okay, so that is the idea, that when the antigen is complexed with antibody on surface of the cell, complement causes the lysis of the cell, and we will going to use this phenomena to detect those antibodies that usually cannot possible to detect or quantify with the help of serology. That means with the help of reaction with specific antigen against those antibodies. Okay, so those antibodies that cannot be detected by reaction with antigens are detected by their function, that is complement fixation, that here we are going to do, right? That means the situation that I have explained, that is how antigen antibody complex will form, and then complement will bind with the Fc portion of the antibody, then facilitates the lysis of the cell. We will going to use the entire phenomena to find out, not only find out, to also quantitate the amount of the antibody and also antigen. Okay, so here the things that are written, I have already explained that antigen antibody reaction lead to immune complex formation. It is nothing new, that produces complement fixation via the classical pathway. Okay, so that is when complement take part in antigen antibody reaction. So that is what I have explained here, right? So suppose it is a bacterial cell. On the surface of bacterial cell there are antigens present, and there are specific antibody for that specific antigen, and complement is bind over the Fc part of the antibody. So there can be a reaction which facilitates the lysis of that bacterial cell. Okay, that is the classical complement pathway. Okay, so when these complexes are on bacteria or red cell, it should be red blood cell. I have just written here red, sorry for that, it will be red blood cell. Okay, now the complement brings about the lysis of the cell involved. Okay, so now, so let me change the thing. Okay, now it is right that it is red blood cell. Okay, and other cells, the complement brings about the lysis of the cell involved. This may be explained. We can use this technique that I have told you, that to determine the amount of antigen or antibody present in the patient sample. And remember that this complement fixation can detect antibody at a level of less than 1 microgram per liter. Okay, so let's see how it can be done with the help of a diagram. So I have downloaded this picture from the internet, you can also find it out. So the complement fixation step consists of two stages. One is complement fixation stage, another one is indicator stage. Okay, so here we will perform one positive test, another one is negative test. Okay, so suppose, let's say here we are considering that both the cases we are considering, we have two patients, we don't know who is infected, we are performing two complement fixation test. So we have taken a test tube for the patient 1, we have added antigens and complement. Now try to think about this. If the serum contains antibody, what will happen? Complement fixation will occur because other two components are present, right? Antigens are present, complements are present. So here antibodies are present, but initially we cannot determine this, right? We are here capable of visualizing that yes, antibodies is present, but let's say we don't know. So complement fixation is occurred here, but we don't know. Now how we can know? For knowing that yes, complement fixation has occurred, we will do a indicator stage where we will add sheep RBC antibody which is specific to that sheep RBC. Okay, now try to think about this. If antibody will not present here, so complement will free, right? So now when we will add sheep RBC and the antibody to sheep RBC to that test tube, what will happen? The complement which was free react with the FC portion of the antibody to sheep RBC which has already reacted with sheep RBC, and as a result complement fixation of the red blood cell occurs. That will change the color of the solution red. Okay, and we will consider them as a negative test. Why? Because remember, as here antibody was absent, that is why complement fixation is possible in the indicator stage. Understood? That is why if your solution changes its color, then make sure that patient has not developed any kind of antibody against that antigen. That means patient may not be infected with that particular disease. Okay, now as here antibody was absent, as here antibody was present, that is why there is no change in the color, right? Because complement was already in reaction with antigen antibody, right? So that is why sheep RBC cannot be lysed, and as a result solution remains same in color. Okay, so no hemolysis, complement tied up in antigen antibody reaction. That is the positive test. Okay, so all available complement is fixed, that by the antigen antibody reaction no hemolysis occurs, so the test is positive for the presence of antibody. Okay, now in another test, that is for the patient number 2, there is no antibody present in the serum, right? So that is why upon addition of antigen and complement, no complement fixation will occur, but we don't know. That is why we have to perform the indicator stage. So when we have add the sheep RBC and antibody to sheep RBC, the solution of the color changes, okay, which is converted into red in color. So as the solution changes its color, it is obvious that it is a negative test. That means patient's serum do not contain any kind of antibody against that particular antigen. Okay, understood? That is the entire concept of complement fixation. It consists of two step: complement fixation stage and indicator stage. After the completion of two stage, if your solution remains the same in color, make sure that it is a positive test, and if your solution turns red, make sure it is negative. Okay, now let's talk about advantages of complement fixation test. So first advantage is you can perform complement fixation test for both bacterial and viral infection all at the same time. Okay, and second one is economical. Okay, but the first advantage is in turn a disadvantage for complement fixation test because it is not sensitive. You cannot do immunity screening on the basis of complement fixation test. Okay, and also it is time consuming and labor intensive. Okay, and often it is non-specific. That is why, as it is non-specific, we are performing bacterial and viral infection at the same time, right? So it is as well as a advantage as well as also a disadvantage. Okay, depending on the point of views. So that was the concept of complement fixation. So I hope this video will be helpful for you. Thank you for listening to this class.